Imaging the thoracic aorta to establish its diameter is essential for gauging the risk of dissection. However, current aortic diameter cut points do not identify all patients at a high risk for dissection, and a more precision-based strategy may be warranted. Accumulation of glycosaminoglycans, or GAGs, is a well-recognized feature of thoracic aortic aneurysms. This is illustrated here by the blue staining material. The purpose of the study was to develop an MRI approach to assess GAG content in thoracic aortic disease. We evaluated 104 ascending aorta samples harvested from individuals with or without TAD. GAGs were quantified histologically based on Alcian blue staining and imaged ex vivo using chemical exchange saturation transfer MRI. This approach capitalizes on the presence of exchangeable protons on the hydroxyl groups of the repeating carbohydrate units of GAGs that effectively generate an endogenous MR contrast signal upon receiving a specific radiofrequency pulse. Control, non-dilated ascending aortas displayed weak Alcian blue staining. In contrast, GAG staining was evident in dilated aortas as either diffuse interlamellar accumulation or large pools. Quantitation revealed a 2.7-fold increase in GAG content in aortopathy samples. Furthermore, GAG content correlated positively with aortic diameter and inversely with elastin content and smooth muscle cell content. To determine if GAGs could be imaged by MR, samples were scanned at 9.4 Tesla. This depicts the T2-weighted scan of a 5 centimeter aorta and the same tissue imaged using CEST MRI. This reveals a territory of increased signal which closely matches a region of GAG accumulation evident by histology. Quantitative data from 25 patients revealed a significant correlation between the CEST signal and histologic data. We next evaluated region-specific CEST signal within a given aorta. As shown, regions of high signal, medium signal, and low signal corresponded to histologic GAG content. Quantification of 18 equidistant regions around the aortic ring displayed a significant correlation between the CEST MR signal and GAG content in registered images. This novel imaging window into the aortic wall raises prospects for aortopathy risk assessment beyond lumen dimensions.